Hi, good afternoon. My name is Gabe Pelletier, and I'm going to discuss today the results of 3 years of invasive species monitoring in Massachusetts for the Massachusetts Department of Conservation and Recreation. I'm going to focus on zebra mussel and the invasive aquatic plant hydrilla. So to begin with zebra mussel was first detected in Massachusetts in Laurel Lake in 2009. Laurel Lake is a area in the Berkshires which is very highly recreated, and there are many ponds and lakes in close vicinity to Laurel Lake, so the risk of invasion is fairly high. In response to that, Massachusetts DCR conducted fairly rigorous conventional monitoring and did not detect zebra mussel outside of Laurel Lake. We were next contracted to conduct annual eDNA early detection monitoring in their high-risk ponds, and we created a sampling plan that was going to focus on areas of high risk of introduction within these high-risk ponds, and we're going to take the advantage of having Laurel Lake as a positive control site to study the factors that were going to increase our ability to detect zebra mussel at our high-risk ponds. First, we wanted to, so I'm going to discuss what we learned by studying our positive control site in Laurel Lake. First, we wanted to determine when to study, and based on our knowledge of the species, we keyed in on sampling during the peak spawning time and post spawning, so June versus September in this case. We found that sampling during the post spawning season was so we sampled the same areas, both in 3 sampling areas, 6 replicates each. We found that sampling during the post-spawning event in September was more advantageous to detecting zebra mussel. Our analysis is that the increase in zebra mussel there's an increase in zebra mussel bioaccumulation within the water body during the post-spawn time period, and therefore that is why we had an increase in positive replicates. Next, the question was, where in the water body do we want to sample? To learn this, we created a sampling plan that included 27 2-liter samples throughout 9 transects dispersed throughout the lake. Each transect included a shoreline sample and an offshore sample that corresponded with the shoreline sample, an offshore surface and benthic sample. We found every single sample positively detected zebra mussel, and there was minimal differences between replicates. What we did find was there was a great variety in the concentration of eDNA within each sample, and specifically between each sampling location. We found that our offshore benthic samples had a much higher concentration of zebra mussel DNA than our shoreline and offshore surface samples. So based on this study, we recommend sampling for zebra mussel during the post-spawning time period in offshore areas near areas of high risk of introduction. In addition to our zebra mussel sample, we've taken on some pilot studies to look at qPCR single species detection for invasive species such as hydrilla. What we've learned by sampling for hydrilla is in 2020 we worked with Nature Metrics and created a primer, and we have positively detected the monoecious strain of hydrilla. And now we've also created, come up with similar to zebra mussel, a seasonal sampling. So we sample the same sites in the growing season and then compare those results to when the species is decomposing and whether that breakdown of the plant material in the water was more advantageous to sample or not. What we found was that it was not. Sampling during the growing season is much more advantageous. Much like any form of eDNA sampling, we found that increasing our number of samples and running a minimum of 6 technical replicates was advantageous to detecting the species and collecting the eDNA. And that's my quick, brief overview of what we've been doing, but if you have any questions, feel free to reach out to me. Thank you.